My name is Hannah Moon. I'm a graduate student in the Porter Vision Lab at the University of Hawaii at Manoa. I'm interested in how seabirds perceive light and how different colors of light affect their temporal sensitivity. Today I'm going to share with you some preliminary results and data from my first field season here on Kauai. Thank you. So my research focuses on 3 Hawaiian species in particular: the newel shearwater, the Hawaiian petrel, and the wedge-tailed shearwater. Populations of the endemic newel shearwater and Hawaiian petrels are endangered and experiencing steep declines. Wedge-tailed populations are considered stable, but populations fluctuate and they're exposed to the same threats as the other 2 species. Ecology, especially the foraging ecology of a species, is known to alter visual systems. Each of these seabirds differs from the other slightly in their foraging ecology. The newel shearwaters are deep water pursuit divers who regularly dive up to 50 meters deep for food. Hawaiian petrels feed almost exclusively on food scooped from the surface of the ocean, and wedge-tails are generally plunge divers who get most of their food within 2 meters of the surface. Seabirds here in Kauai face 2 main anthropogenic threats. Juveniles are threatened by coastal lighting infrastructure. They're attracted to street lights where they land under lights on roads and are subsequently hit by cars or killed by predators. The adults have a much lower attraction rate to lights, but they frequently strike power lines when transiting to and from their burrows at high speeds in the dark. These strikes frequently injure or kill the birds. The mitigation of these threats has included manipulating the spectra of lights around power lines and lights. So every color under the sun has been proposed to conserve birds. Behavioral studies of response to color and intensities of light are largely inconclusive or contradictory. Still, the color and intensity of light has been changed near seabird colonies without scientific evidence or assessment of its effectiveness. Green has been arbitrarily chosen to design lighting systems aimed at conserving seabirds here on Kauai. So green lasers, as evidenced by this picture right here, they have been successfully used to reduce adult seabird strikes on power lines. However, it's not known if these lasers are efficiently targeting adult seabird visual systems. Use of green light to reduce juvenile light attraction here on Kauai has resulted in both anecdotal reduction of light attraction events and one of the worst, second worst, I believe, light attraction events on the island that affected both juveniles and adults. We need to stop guessing and design seabird-friendly lighting systems, and we want to know, do the seabirds on Kauai respond differently to different colors of light? Are there differences in response to light in the retina between adults and juveniles, or is adult avoidance of human lighting systems learned? This leads me to my hypothesis that the observed difference in light attraction between adults and juveniles is due to physiological changes in spectral sensitivity at different life stages and between species. To test my hypothesis, healthy birds were rescued by Save Our Shearwaters and then selected opportunistically. All the birds were grounded by light attraction during fall of 2018. And I want to say thank you so much to my co-author Tracy Anderson and the Save Our Shearwaters crew for making this work possible, despite working with very rare and often very uncooperative birds. Birds were dark adapted for 30 minutes prior to anesthesia and testing. Once they're asleep, the bird's placed on a towel over a heating pad on my metal table right there. And then right here, that is my flashing LED stimulator light, and that's positioned over their eye. I then test the bird's response to different colors of light using electroretinograms. So electroretinography measures the strength of electrical responsive cells in the retina. This gold electrode is placed gently on the cornea, and using that I can measure changes in the eye as small as 1 microvolt. Then I flash a light at increasing rates to determine the flicker fusion frequency. Flicker fusion frequency is defined as how fast a light can flash before it's perceived as a constant light. It's measured in hertz. The faster a light can flash, the higher the temporal resolution of the eye, meaning it is easier to detect and resolve motion. Birds have the highest flicker fusion frequency of any vertebrate. They can detect flicker at up to 140 flashes or hertz per second. To put that in context, movies look like a continuous motion to humans when each frame is as slow as 24 per second. I chose to test the flicker fusion frequency and strength of response based on known aspects of spectral perception. So this is a very generalized model of spectral sensitivity in the 4 cones of violet-sensitive birds like the wedge-tailed shearwater. Hawaiian petrels and newels have not been fully characterized yet. Seabirds in general are known to have a violet instead of an ultraviolet sensitive system. They have a peak sensitivity of about 400 nanometers in the violet photoreceptor here on the left. And that doesn't mean that violet sensitive birds can't perceive ultraviolet light, which is underneath 400 nanometers. Even humans can see down to 380, we just don't see it very well. So birds were selected for testing in one of 2 experiments, either UV light in 5 intensities or 3 intensities of blue light and then 3 intensities of white light. UV is a wavelength of interest to conservation efforts. Blue light is of interest as 2 of these species forage underwater where blue light is most prevalent. and white light covers the broad range of spectral sensitivity. The response is then visualized in peaks that look like this. The strength of the response is represented by the black bar and quantified by the height of these peaks. The flicker fusion frequency is determined when these peaks flatten out and no longer measure above background noise. And that means that these birds are perceiving a constant light. 18 total birds have been tested so far. So this table shows how many juveniles of each species and with what colors of light I've tested. The asterisk represents the single adult that I've been able to test, which was a wedge-tailed shearwater using blue and white light. Blue and white lights were tested within the same bird to make efficient use of time and to increase the number of samples. And these are my results. So when flicker fusion frequency is plotted against increasing light intensity, it is clearly grouped by color and increases with intensity. This is data for all the birds that I've tested. The flicker fusion frequency did not change between species or between juveniles and the adult. Each bird processed each color of light at about the same speed. So white light has the highest flicker fusion frequency, averaging between 60 and 70 hertz, and one bird tested almost as high as 90. Blue is in the middle, around 50 to 60 hertz, and UV was the least sensitive with an overall flicker fusion frequency at 25 to 35 hertz. So if we shift now to looking at the strength of the response, just the general height of these peaks, this is the strength in microvolts plotted against the increasing light intensity in all birds. So UV response, once again, is also the weakest, ranging from about 5 to 15 microvolts. Unlike white and blue, the response strength doesn't increase substantially with brightness. And this indicates that all tested birds likely use UV solely for enhancing visual discrimination. So how does the strength of this response change between adults and juveniles? So only one adult wedge-tail shearwater has been tested so far, but it's interesting that it had a pretty weak response to light. The solid line represents the adult shearwater's response to blue on the left and white over here on the right, and it's plotted against light intensity. And the maximum response to white light measured about 15 microvolts. The dashed lines here are the response of the 3 juveniles I've tested, which had a clear increase in response to light intensity and a range of response between 10 and 100 microvolts. While more data on adult seabirds is necessary, the adult clearly has a weaker response to light. This is consistent with the observation that light attraction is mostly seen in juveniles and not the adult birds, which cross over coastal lights every night during the breeding season. So how does the strength of response vary between species? So some key differences emerge when strength of response is plotted against light intensity by species. Hawaiian petrels aren't plotted here because I've only tested one of them with blue and white light, but the response so far has been very similar to that of a wedge-tail. Newells have a stronger response to blue light, averaging around 50 microvolts. And wedge-tails on the right have a stronger response to white light, also averaging around 50 microvolts. The average response to light in these two species is nearly inverse. So this matches predictions based on ecology. The newells are deep water foragers, catching fish where long wavelengths of light from the sun are not present. These data indicate a shift in light perception towards shorter wavelengths of light, very similar to the shift in spectral perception towards blue and green seen in deep water foraging Humboldt penguins. So in summary, my hypothesis is supported. There is a physiological difference in response to the color of light. All species have a weak response to UV light, indicating a violet-sensitive visual system for enhanced color discrimination. There is evidence that juvenile birds have a physiological response to light, a stronger physiological response to light than adults. And newel shearwaters have a stronger response to blue light, indicating a shift in spectral perception towards shorter wavelengths driven by their differences in foraging ecology. So what does this mean for preventing seabird deaths here on Kauai? Due to the low response of the retina in all tested birds, pure UV light will not be an effective management tool when designing seabird-friendly lighting systems. While further tests are needed, this shift in spectral perception towards short wavelengths indicate green light may be targeting spectral peaks in seabird vision, particularly in the newel shearwater. This is good for lighting up power lines using the lasers to save adult birds, but it could have negative implications for using green light in outdoor areas that attract seabirds. So how do we protect birds on Kauai? In 2019, I'm hoping to test more adult birds, and I would like to try to isolate testing the sensitivity of all 3 species to red and green light. I'm also working on collaborations focused on creating a full model of seabird vision using molecular investigations of peak photoreceptor sensitivity, spectral transmittance of the properties of the eye and retina, and some retinal ganglion cell distribution counts to determine spatial resolution in the target species. Ultimately, we want to use this model to design seabird-friendly lighting systems with spectra that do not attract juveniles. To aid in this effort, if you or someone you know works in seabird rehabilitation and would be interested in donating eyes to research, please contact me. And with that, I'd like to say thank you. Thank you to Tracy Anderson and Save Our Shearwaters and the Kauai Humane Society for making this possible, to my advisor Megan Porter and the Porter Lab and the University of Hawaii, to my co-author Mark Travers and the Kauai Endangered Seabird Recovery Project, to the Kauai Island Utility Cooperative for funding my work. And with that, I'll take any questions.
And we have 5 minutes for questions, so I think
Yay.
they're going to be a lot.
I was expecting that. So I saw your hand first.
Yeah, I wrote it down. Is the strength of the response of these birds contingent or influenced by the order you present the stimulus? I don't know anything about vision, but with hearing, you can have, like a partial threshold shift. If you have a really loud noise, then you can't hear as well later. And so I'm wondering if that is true, or anything like that is true for eyeballs.
Absolutely. So some of that can be affected by order. Between every test, there was a break where there was a dark period between every single test, and all birds were tested in increasing flashing rates and increasing intensities, using a similar progression with all birds.
Cool, thank you.
Yes.
So I also don't know anything about vision. Why
If anyone, just don't mean to interrupt you, but if anyone wants to learn about vision, that's what my lab focuses on, and I can tell you all the nitty-gritty details if you catch me outside at some point. But please.
So what would be the mechanism by which juveniles would, or how adults would become less, sensitive to light?
So I actually, I only have ideas, but there has only been one ontological study focused on juvenile seabirds and their eye development, and it was on leeches, storm petrels. This is Mitkiss, Nevitt, and Kelber 2017. This is a map of retinal ganglion cell distribution. So I didn't study this, but retinal ganglion cells are where all the information from the photoreceptors I did study, it's where all that information is summed up. And so retinal ganglion cells do change as the birds get older. So the juvenile birds, let me start with this. So the adult birds all have a very similar eye layout, so like the one on the right. They have a very clear visual streak with a high density of ganglion cells. And juveniles kind of have very spread out ganglion cells, their visual streak is poorly developed. And even in birds that have just left the nest, they don't really see very much consistency in how developed the eyes are. So the working theory is that birds that live in burrows, their vision hasn't fully developed by the time that they leave.
Yeah, I think you're spot on. I think it's a question of thresholds, and I think we've probably been using the wrong word. I don't think it's attraction. I think it's just confusion. I think these birds...
I have a lot of my own theories about that too, sir.
It's like when you come, when you go and see a movie and then you come outside, you're often blinded. So I think these birds are entering, they have poorly developed vision. They're entering these areas with bright lights. They become confused and they can't leave, because when they do go into the dark areas, they can't see properly because those thresholds have been crossed.
Exactly, and I don't know if any of you have ever been up and seen the moonlight on the water during a time when the birds are less attracted to lights. It's if you climb a mountain, especially around this time of year, the moon on the water is very bright and it's a very powerful signal, and the birds don't have that when there's a new moon.
I'm just trying to put all this together still, but this idea that the very young birds have a poorly developed reception system for light and poor vision, perhaps as well, is it just even more interesting that they're able to imprint perhaps on the night sky and remember where they might be from and return to that place with such a poor developed perception.
Yeah, seabirds, they do have a very sophisticated sense of smell starting at a young age. So while they can't necessarily smell their burrow from far away, I'm getting back to the colony probably has a lot to do with that. In the back?
So this is an issue that I've been trying to understand. We can look at, and you're looking at, what the physiological response is to light. How do you make the association from that physiological response to the behavior that we're observing and dealing with all the time? Where's the connection?
So that is going to require more experiments. I'm trying to use this as a basis for further experiments to design an actual model of their spectral sensitivity. And using those models, we can actually design better behavioral tests. Right now, people are just implementing colors based on ideas they've heard, and no one is testing them, and no one's actually looking at what's really working. So I feel like we're just taking this back to the baseline right now, trying to figure out what's most important to look at before we actually get back to that behavioral testing step.